So good afternoon, and nice to see you all here, that you managed to come here after this long break. Thanks, Nico, for introduction. So my talk will be more like a little bit like a story, how we have kind of done quite a long time of work on collaboration with stem cell scientists and especially on story about IPS-derived cardiomyocytes and how we have kind of combined in silico and in vitro. And let's see whether this works. Okay, no, that's a small delay. First of all, we actually something like 60 people in University of Tampere, part of this kind of Center of Excellence in Body on Chip research, and done even longer than this, work together, 6 PIs, 60 people, really spanning different areas from stem cell sciences, biomaterials, microfluidics, and myself, who is more like, in a way, computer science background, I would say more. And my lab is really doing in computational modeling, biophysics in silico modeling, you could say, also electrophysiology, biomechanics, and being part of this, we are doing work in various cell types, but this story is really about cardiomyocytes. And there's a couple of reasons for that. First is that cardiomyocytes is really, I would claim, that the organ and cells that are mostly modeled. None other cell type is so well known actually on the basic functions as cardiomyocytes. And this is my kind of motivation intake on kind of personalized medicine. So we have these two areas that always people refer, like personal data, gene RNA, other proteomics. But then one area is computational models, and of course the fourth one is the MPS that we all are here for. So I would like to kind of claim that all this is needed for future personalized medicine. And I think most of us here agree very well. I don't need to stress it too much to really get to the patients. If you think about the modeling of human physiology, of course, the tradition has been that we have the preclinical animal and population level models for humans. And now we have the MPS really emerging. But traditionally, the modeling has been really on looking to kind of, I would say, the organ level, human level modeling, and a lot of work has been done on there. And now with the MPS systems, it's been opening us for us modelers a really nice way forward to look in the cell functions. Because nowhere else we have had this kind of nicely controlled system for computational modeling. We need data. And those of you who are making these MPS are kind of treasure trove for us to give the data so that we can learn more about the cell functions and have nicely controlled input-output relationship that we can then convert to computational models. And I hope that all this will lead that we can kind of close the loop as well and go back to the patient, the modeling on whole body level and maybe to the patient itself. Just a little bit about the in vitro side. So we have actually worked a long time with iPS-derived cells. One reason is that Professor Aalto-Setälä, our cardiologist, was actually in California in the same lab where Professor Yamanaka was at the same time, and we got those cells quite early. But this just kind of, the video there just shows that beating cardiomyocytes, and of course you can add drugs and have the ECG of the cells, action potential derived field potentials and see how it changes with the drug. You can also see, of course, that they are beating. So there's a contraction there, and that's one thing that I would like to address later. And just about the data. We need data for modeling, and if you would like to model the cardiomyocytes, you would like to get electrophysiology. Calcium is important as well because it drives then downstream the contraction. If you get the contraction, we have a kind of nice assay, and we have actually worked quite a lot on that. And this one example of paper where we show that you can actually drive all these three assays or measures simultaneously from the cardiomyocytes in vitro. So we have the data. And just kind of importance of this is that, for example, in certain genotypes, the effect what is happening there on mutation is could be ion channel level, it could be calcium level, or here it actually on the action, on the contraction level, where the contraction mechanism is affected by this mutation. So if you try to model these, of course, these are the levels of modeling as well. Just to show that what the in vitro world is going on in Tampere, so we are really actually looking more on injury and ischemia and try to look for that. But anyway, it's kind of nice that we have these disease in vitro and you can model and look the mechanism as well. And of course, the question is it valid for the patient? And in a way, that's been here many times. This is the cells in vitro might not be really the same as in patient. Neither they are in cardiomyocytes. They don't have the structure. They lack certain kind of, let's say, mechanistic events. Some ionic currents are not the same. So really, sorry about this quite complicated slide, but this is a nice slide made by my postdoc, Jussi Koivumäki. And idea is that from the patient, we can, of course, get a lot of data. And traditionally, it was made for whole body modeling. And IPS, we get a lot of insight on how these cell works of the patient cell works, how the calcium contraction, action potential, other works. And to really close the loop, we need these IPS-derived cardiomyocyte models. They are not the same as real cardiomyocytes, but these are really the thing is that we are making model of the model. Your in vitro model turned to the in silico model. And if we have that, then we can really look inside the mechanistic things. Hopefully we can translate to the patient. I think, to my knowledge, nobody has really done this whole circle. The pieces are there, and hopefully in future we can. But just think about what these models are in silico. So of course they try to replicate the cardiac function, what we see. And if you think about electrophysiology, there are really a bunch of differential equations. Like here is one exchanger. Sorry, here is the color. There's this exchanger, and you have the equation, nice equation there. And from all these, of course, you have another equation. A lot of parameters, a lot of equations. But these really define the electrophysiology event, and they can produce these kind of action potentials. And nice thing about that, of course, is that we have all the data, all these ion currents, all these opening and closing of ion gates dynamics, and we can tweak these and produce new set of models from different diseases, like for the different, let's say, like long QT is here example where you have prolonged action potential. We can add drugs, so changing these ionic currents, because a lot of drugs are really meant to change the ionic mechanism of cardiomyocytes. So this is a nice tool to play around. And I think we actually started this business on IPS-derived cardiomyocyte modeling. Michelangelo Pacchi came to my lab as a PhD student and later came as a postdoc, and his model, I think, is one really first one on this. And how it came to be is that some people have really done painstaking work and measuring these ionic currents from IPS cells. So a lot of data was needed to make the model. And then Michelangelo really fit the data to these current traces and make the model. And so we had the model, and we could kind of show that in a way it really produced the same stuff that you can observe in vitro. And there's actually, this is an adult model that has been well let's say characterized. And these are two different models, ventricular and atrial IPS-derived cardiomyocyte models. And we were able to see what is the difference, how the ionic mechanisms are different, whether the drug effects were the same. We observed that it was about the same effect. Some cases, the IPS-derived cells actually have bigger effect of drugs than adult cells. So they were more sensitive. So this was quite reassuring that they were quite good models. And of course, this was very nicely balanced. The same things have been observed also in vitro. But of course, it's kind of boring. You have a nice model, but it's just like having one cell in your dish and saying that now we have a proof of concept. And how to go forward really was think about how to go this population level, high throughput in silico. And what we did, what Michelangelo did, was to make thousands of models, tens of thousands of models. And you can do it many times, many ways to kind of build this population and this kind of brute force, small changes in all the parameters, and you can get a lot of huge set of models with different kind of behavior. And like here are the action potentials of all these thousands of models seen there. Of course, they might not be the correct ones. How we select the ones that are not physiological. What we did was to we went back to the data. It's available. So all these boxes here are from different data sets measured from in vitro, and different biomarkers of action potential observed. And we selected those ones that are outside this and omitted those. So we pruned the population and took only those fit to all the biomarkers measured in vitro. So we generate this kind of nice set of models. And for example, it's kind of interesting effects start to happen because you could really have a population modeling and have population drug effects. So like this kind of small example where you have this hERG channel block, and in some cells, it really produced the effect that was expected. Some produced arrhythmia, and actually some of our cells died. Luckily, it was actually in computer, so nobody was hurt in this experiment. But of course, now we could actually look back and see what these models are and then go back in vitro. Unfortunately, we haven't really done it properly, but that is now. We can look this population and see what other kind of cells that are in danger of having arrhythmic or some other effects. And interestingly, we did also this long QT study on this and producing population of long QT. And when you add this mutation in your cells in silico, we observe that we have actually there two populations. We have populations that are sick and populations that are not sick. So we have kind of patients that are asymptomatic and others are not. It's like in real human population as well. Not all who have the mutation leading to long QT are actually long QT patients. And now we had actually had drug and test the drug with asymptomatic and symptomatic population. A little bit complicated to read there, but so we have pre-population control. Then we have this mutant asymptomatic population. We add drug. There's not very much change, but when you have a mutant symptomatic that has the long QT and you add the drug, you really have a clear change there. It's quite nice results on considering that this was really in computer. And of course, it's quite hard to think that we have the original model still the same one that we derived from the traces of ion channel kind of opening and closing. So we thought how to make a new one or how to let's say make it easier. And this is one attempt that we just take the traces of action potential calcium and use those biomarkers and tune a new model based on just those. So we didn't have any traces of real gating of different ion channels. And we were able to make this. And this kind of lead to us to really try forward and think that can we make these kind of things purely on data from in vitro. And we were able to join forces with with Emilia Incheva. She had done quite large still, even modern standards, quite large set of data from, let's say, 170 multicellular samples, 200 cells at least each, and they had traces of calcium and voltage from there, and they have tested also 12 compounds. Problem was that they actually did this all work in room temperature, and all our models were really for the human, and they were on 37 degrees. So first task was to trace or transfer our 37-degree model to room temperature, and it was actually one kind of example how robust these models actually are. We were able to do it by really changing the gating variables based on literature review on how the temperature is affecting the different ion channels. And based on this, we were able to actually make nice set of populations and test these 12 compounds and get surprisingly similar results with our in silico models that Emilia and others have made in vitro. And in addition, they were quite interesting that we were able to see similar behavior. So we observed kind of individual models that produce early after depolarization, similar things or similar kind of amounts that you could find in silico or the depolarization failures or irregular rhythm. So in a way, the population in silico and in vitro were surprisingly similar in many ways, not just respond to drugs, but this kind of, let's say, I would say, kind of personal traits of, of the cells. So in principle, what we can do is to have these cardiomyocytes, have the traces of calcium voltage, hopefully also the contraction, and have this kind of population model idea and transfer these data that we have measured to populations of models in silico. And of course, these models here can be certain phenotype. They can be individual. So we could have 10 wells or then MPS systems from individuals, because also our cells are not the same. They have certain variability. And we can tune these populations depending on those populations there. And of course, using these, we can then test all kinds of things. We could maybe test some drugs, toxicity, or certain conditions, whether it's ischemia or something else, and then find the populations, find the subset of cells or disease or drugs that are important, and really go back then to the bench. And if you think that this is kind of hopefully the kind of way to speed up things, they are not competing, both are needed. But we all know that if in a way, MPS systems are nice, they're excellent, but going really high throughput is difficult. And testing number of population, number of different individuals and number of compounds is a lot of work. But this is, of course, we haven't fully tested, but maybe something that hopefully we can prove later on that this really can be done. So having the MPS on the pipeline or the in silico MPS on the pipeline of MPS testing. Okay, just to kind of show something where we're going now. Of course, these models lack the contraction, and this particular work here is by Amin, and it's under review. But you can, if you Google it, you can find it. It's online, as normally your papers are online earlier than they're accepted. And here we really integrate electrophysiologically and biomechanics, so contraction of cardiomyocytes, and also energetics, because energetics is needed, energy is needed for especially for contraction. And having this, we were able to actually look the new HCM drugs and get an insight on how they work and how they affect this. I hope maybe next time I can open this more. But just to kind of show, this is a list of papers and others that was actually data used for making this model and validation of the model. So to make this and validate what we did, needed a lot of work by many people in their labs in vitro. So to be able to do this work, we need really a close collaboration. We have a close collaboration in Tampere with these guys, but sometimes it's not enough. We really need kind of community effects to pull these together. Okay, and of course there's things to do. It's a nice heart in Katrina Alseta's lab. They try to do a kind of flat 2D culture, but sometimes the cardiomyocytes decide to do something else. Of course, this is what we would like to get, nice cardiomyocytes that are not just cardiomyocytes, because heart is not cardiomyocytes. It's I don't remember the percentage, but I think something like maybe even 50% or something like that. There's all kinds of other cells. So having just cardiomyocytes is not enough. So really to kind of think about how we can really bridge this gap from our cells to there on whole heart level and patient level, and really have the feedback there. This is actually also my lab. It's rat heart from microCT. And this is our attempt to really see the contraction on 3D cultures of iPS-derived cardiomyocytes. So I know it's interesting to see here how much work is now doing on this direction. And of course, our modelers, we need to follow this and see how we can cope, because this will bring a lot of complexity also on our modeling. More data needed and more collaboration. Just pointing out one interesting paper that just appeared by this Hubech and others. There's actually a couple of names from our friends from Norway, Simula Lab, that are also doing in silico modeling. And what they actually did in this paper, they managed to make, by having really nice MPS system, but also this metabolically driven environment, to make more adult-like cardiomyocytes. And what they did also in silico is actually they took one of our models from Michelangelo and make it more adult-like by getting the traces of some traits from their cells. And one interesting was actually the size of cells, because adult cardiomyocytes are bigger. In fact, surprising a lot on electrophysiology, but also some calcium handling. But they were able to kind of show that modeling can really explain what is happening in these more adult-like cardiomyocytes. So anyway, virtual precision medicine, I think this is what we do here in vitro body on chip MPS. It's going to play an important role in here, but I think we need also the data science approaches. I think in this, look forward to hear more of this, and also the modelers to kind of connect the dots and connect the different kind of areas. And just to thank these guys here who did all the work, Michelangelo, Paki, especially Jussi Koivumäki, Antti Ahola has done a lot of work in vitro, and Mohamed Amin, and collaborators, especially from Bologna, Oxford, and Washington. And I thank with that. And if somebody is interested to hear more about computing and cardiology, please come to Tampere in autumn. Thank you.
Thank you very much. We have time for one question.
Hi, this is Ishan from Berkeley, actually from Kevin's lab. So I had a question regarding your scaling from immature to mature cardiomyocytes in the model. Now, in the Ashlag's and Kevin's paper, they assume the functional invariance of the proteins and scale the number of ion channels based on just the size of the cell, right? Is your scaling also in a similar vein, or I didn't understand how that scaling was done in your model?
Yeah, of course, it's what happened in that last paper that it's not from ours. I think that is really what they did was looking at the size, looking at the kind of number of ion channels that is really different in adult, and that is of course what we also observe. Of course, the kind of basic function of ion channels is very similar as whether immature or mature. The ion channel structure, protein structure is the same, but how many there are, how they're connected, and especially when you then start to look at the kind of how it's connected to calcium handling inside the cells and how it's connected to the, let's say, biomechanics of the cells, that is quite a disarray on these immature cells.